8 or 12 or something.
Yeah, salmon are polyploid. Some salmon have 4 copies of every chromosome. Do they do mitosis? Do salmon fertilize and then grow bodies? Yeah. Those are tetraploid cells. Those tetraploid cells do mitosis. It looks different than diploid cells doing mitosis. It's the same process. So restricting mitosis to somatic cells, yes. Ploidy, probably not so much. So what cells perform meiosis? Gametes, sperm and egg, those are the products. That was what the question asked. What is one name for a type of cell that is produced after meiosis? That's gametes, sperm and egg, which most of you put down. So that's a good start. Mitosis and meiosis. Today we're going to look a little bit more at the details of meiosis in particular. Oh, and before we get there, especially for those of you that have downloaded the slides and are looking at them, you might not believe it by looking at me, but I'm the advisor for the Society for the Advancement of Chicanos and Native Americans in Science. I've been involved with SACNAS for decades, actually, and I'm currently the campus chapter advisor. So I'm here to tell you particularly about, they hold events all the time, but this one's of particular interest to this class. Did you know that we have a Department of Justice crime lab on campus? We do. They don't really interact with campus because they're a crime lab, so they don't really interact with other parts of campus. But we do have somebody from the DOJ crime lab that's going to come talk to the SACNAS chapter about forensic science. I'm not sure she's doing genetics forensics, but there's a chance that she is. And so if you want to go, it's free. I suggest that you go if you want to. And if you want, use that QR code to sign up. They might be providing food, which might be why they want to take attendance of people that are interested in attending. But you're welcome to show up even if you don't RSVP. Now, back to mitosis and meiosis. So meiosis, both of these processes start with, let's talk about human, since we're all humans. What's the ploidy of the cell that starts either of these processes? What's the ploidy of the cell that will start either of these two processes? Diploid. Diploid cell, 2N. Okay. So far, mitosis and meiosis are exactly the same, because there's a cell in the gonad that divides and the daughter cells become the sperm and the egg. But that initial parent cell is a diploid cell, just like all the cells in our body. Next step, DNA synthesis. So in preparation for cell division, chromosome number doubles. Every chromosome duplicates. So then we get a 4N cell. Temporarily. It's not tetraploid because we already talked about ploidy is determined at G1 during the cell cycle, not at mitosis or meiosis. We just got one cell temporarily that has 4 copies of every chromosome. Okay. Next step is the same too. What happens next? After the cell duplicates its chromosome content, for both. At this point, in the next step are the same in mitosis and meiosis. What happens next? They divide. Mitosis, that's the only division. There's a cell division that gets you back to 2 cells. That 4N, 4 copies of every chromosome, divides into 2 cells, 2 copies of every chromosome per cell. And that achieves what we talked about the last time we talked about mitosis, which is you've gone from 1 diploid cell, 2 copies of every chromosome, to 2 cells that have 2 copies of every chromosome. Mitosis. Somatic growth. So meiosis then, how does it differ? 4N cell divides. So it produces, just like mitosis, 2N cells. So what's the critical difference? I think most of you know this. What happens next? they divide again. Meiosis has 2 rounds of cell division because we want to wind up with haploid cells. 4 cells, each of which have N, 1 copy of every chromosome. And we talked last time about why that's important, because when sperm and egg meet, we want to develop an embryo that's diploid, just like we are. We're creating a new human, for example. It needs to be diploid, so we have to have haploid gametes to make, when they fuse, a diploid organism. So that we talked about last time. And I told you last time that I had done something slightly misleading when I drew this, and I will always tell you the truth when I'm lying. I will tell you I'm lying. And I was lying when I drew this because there's a subtle difference that we're going to get to in just a couple minutes. Does anybody know why this is not always true? I drew it a little bit misleadingly. Meiosis. We'll see when we get there. All right. Chromosome segregation and meiosis. How do the chromosomes decide, or how does the cell decide which chromosomes go into which cell, both of the divisions? This is probably something that some of you have anxiety about. You may have seen this before, you may not have. So I want to walk through this. It was already in one of the videos, but I've taught this class enough times to know that I should probably do it again. So we have a 2N cell. This is how mitosis and meiosis starts. And I'm going to draw 2 different chromosomes so that we can see how 2 different chromosomes will decide which cell to go into at both of the cell divisions in meiosis. So if we're doing this, I suppose I should probably color code them. So I'll use blue vertical lines to represent 2 chromosomes. And there's, as always, let me zoom in a little bit. Don't watch if you're seasick. Zooming. There's a paternal version and a maternal version. And then there's going to be a second chromosome that I'm just going to draw as shorter, different length chromosomes. So that's the second. So call them chromosome 1 blue and chromosome 2 red if you want to. This individual inherited 1 copy P from parent, or from father, paternal, and 1 copy from mom, maternal. Typical diploid cell, this is ploidy, 2N equals 4. So, typical cell, it's going to divide. What's the first step? Cell wants to divide. G1 phase. The next step in the cell cycle, synthesis. It has to copy all of the chromosomes to make a second set of chromosomes to provide both of the daughter cells with chromosomes. So what does this look like after synthesis? How many blue vertical lines are we going to have? How many copies of chromosome 1? There's going to be 4. How many paternal copies and how many maternal versions? 2 each. So there's going to be a paternal version, and let's say, just for the sake of saying it, that this is post-synthesis now. So S is that arrow, synthesis phase. Now the cell has doubled all of its chromosomes. It's replicated them all. So let's say this is metaphase, where we can actually see the chromosomes as objects. We've got 2 paternal versions of chromosome 1, 2 maternal versions of chromosome 1, and 2 of paternal chromosome 2, and 2 maternal chromosome 2s. So temporarily we have 4 copies of every chromosome, which is nomenclature that we call 4C, 4 copies of every chromosome. What are the 2 blue P's? I'm not talking about Mendel. Those are green and yellow and wrinkly and round P's. What are the 2 blue P things? There's a particular term for those 2 things at mitosis in metaphase we've already talked about. Each of those 2 double helices. Say it loud, say it proud. Sister chromatids. These are the sister chromatids. So there, for example, are two sister chromatids. The two paternal, the copies of each chromosome, are the sister chromatids right before cell division. This is metaphase, let's say. So we have four different sister chromatids here, four different pairs, rather, of sister chromatids. So the question becomes, if the cell is going to divide, and let me draw a cell right before cell division, those chromosomes have to orient themselves in a particular way so that the two daughter cells get one copy of each. So let's say the dashed line down the middle is where the cell is going to divide. The cleavage furrow, cell membrane is going to form there. How do we arrange these four pairs of sister chromatids relative to that dashed line so that we get the right chromosomes in the right daughter, the chromosomes in the correct daughter cells? This is for meiosis.
Don't they just line up side by side in the middle?
So you could diagram it like that. That's how the chromosomes align. They pair with each other. You may recognize that terminology. So pairs of sister chromatids pair with each other. Pairs pair. Okay, so meiosis. Cell division is going to happen. Let's label these blue and red things with the P's and the M's. Blue chromosomes, which ones do you want to have on the left? The sister chromatids from dad, paternal, or mom? Doesn't matter. Pick a letter, P or M. P. It is afternoon, so you could pick either one, it would be fine. But dad joke. Okay, so if the paternal versions are on the left side, then the maternal versions have to be on the right side. Here is one of the most important things for class this term. It will help a lot when we get to Mendelian genetics. Where do we put the P's and M's for the red chromosomes? Or does it matter?
It does not matter, right?
It does not matter, right. The chromosomes don't know where they are in the cell. They just pair up. The pairs of sister chromatids get together with similar sequences. That's how the chromosome pairing works. Chromosomes with similar sequences get together in that tetrad. The four double helices, the two pairs of sister chromatids, forming the tetrad. Right, so here's a tetrad of double helices, two pairs of sister chromatids. They don't know which side of the imaginary dashed line that doesn't really exist in the cell they're on. So it doesn't matter which side we put the P's on or the M's on for the red chromosomes. Which way would you like to do it today? What do you want to have on the left side, P's or M's? Okay. The critical point here is it does not have to be this way. We could just as easily have written the M's, the maternal versions of the second chromosome, on the left side. But we'll go through the example like this. So then we do division 1. So we've got the long versions of the blue chromosomes and the short versions, the red chromosomes. Now we have two cells that are 2C. Each of those cells has two copies of every chromosome, two copies of the blue chromosome, two copies of the red chromosome. We're doing meiosis, which means we need another round of cell division. So each of those will divide on its own. And I stopped writing the P's and the M's. You can always look up at the top to see where they're going. So what happens after cell division now? Each of those two cells divides. And how do the chromosomes segregate, divide themselves into these two pairs of daughter cells, the four gametes? At this point, the previous image up here at the top, you'll notice the sister chromatids stayed together. Do you during cell division. The two dad versions went into one daughter cell. The two mom versions went into the other daughter cell. So first division, sister chromatids stick together. It's the second division where they get pulled apart. So you wind up with cells that have one blue. Each cell gets one copy of chromosome 1, one copy of chromosome 2. Each cell is C, has one copy of every chromosome. It's haploid. So that's chromosome segregation and meiosis. The only real trick here is to remember that it's the first division where the sister chromatids stick together and go into the two initial daughter cells up here. And then those, so these would be the 2P versions, for example. Make this totally explicit. And then it's the second division, division 2, where those chromatids separate. Any questions about this whole process? Yeah.
Might be a little off topic, but I read somewhere that most of your mitochondria, if not all, is passed on from your mother. Like, how does that work?
Fantastic question. A topic near and dear to my heart because that's what my research lab studies is mitochondrial segregation. But those chromosomes are not in the nucleus. So we're focused, all of this, which I didn't say, so you're right to ask that question, this all happening in the nucleus of the cell. These are nuclear chromosomes. The mitochondrial chromosomes in mitochondria, the organelles, what do mitochondria do? Mitochondria are the fill in the blank. Mitochondria are the powerhouse, which means everybody learned from the same textbook I learned from. Mitochondria are the powerhouse of the cell. They make ATP, cellular energy, that we all need to grow and do these sorts of processes because most enzymes require ATP to do their jobs. And mitochondria are inherited almost entirely from your mom. And actually, we might remind me if I don't respond to your question more carefully by the end of class today, because we're going to be talking about why, in a very tangential way, we can talk about why that is. But yeah, textbooks say we get all of our mitochondria from our mom, despite the fact that in our nuclear chromosomes, we get half from our dad and half from our mom. You get all of your mitochondria just from your mom. Any other questions or concerns about this process? Again, the main point, if anything, is that at this step, right before the first division, the paternal and the maternal versions could be on either side of the cleavage furrow, where the cells are about to divide. It doesn't have to be the two paternal versions going into the same daughter cell. Oh, interlude. Time to engage a little bit more, in case you're disengaged at the moment. Say you're a scientist and you discover a new species. I know hypothetical questions are kind of lame, but let's play it anyway. Say you're a scientist, you are a scientist, you discover a new species. You look at the species, how do you know which ones are male and which ones are female? We're talking about biological sex, not sexual preference or orientation. We're talking about biological sex, not gender. You've got two organisms. Which one's the male and which one's the female? What's the biological definition of the sex? Not for humans, but for any random species you might encounter. If you wanted to know what sex it was, animal, not plant, let's say. It's partly the size, it's almost entirely the size of the gametes. More importantly, it's two things. It's the size of the gametes and their mobility. They tend to go together. The smaller gamete tends to be the one that is mobile. What kind of a gamete would that be? Sperm. Right? Human sperm, flagellated, they're the ones that move around. And the human oocyte floats down the fallopian tube, and the sperm all swim to the oocyte. That's the biological definition of sex. The organism that produces the small, numerous, mobile gametes are the males, and the females are the ones that produce the larger and less motile gamete. The concept here, you don't have to remember this, but this is called anisogamy. This is only true for species that are anisogamous. That just means not same-sized, not the same-sized gametes. There are some species that are isogamous. The gametes are the same size, and then it's kind of hard to define who's the male and who's the female. But this is how biologists tell the sexes apart. It's the definition. All right. This is directly relevant to what I've been alluding to now for a little while, that I lied a little bit or misrepresented something about meiosis. There's a difference, a really important difference, between meiosis in males and females. Do you know what it is?
Is it that the oocyte doesn't contribute the organelles, or sorry, the opposite of that?
Right. So one critical difference is because of anisogamy, because the sperm are so small compared to the oocyte, that's one reason why we get our mitochondria from our moms, is that the oocyte is huge. I don't know if you've ever seen a micrograph. I suggest Googling it, but only when it's a place where it's okay to look at not suitable for work NSFW images, because when you work in, I used to do research in sex determination, so I'd have to do internet searches on all sorts of things related to search topics that had to do with sex and hermaphrodites and all. Anyway, you probably understand where I'm going with this. So you could search for an image. There are great images of the relative sizes of human sperm and eggs. If you don't know, it's really outstanding how huge the difference is between the size of a sperm and an oocyte. So that's the reason that we get our mitochondria mostly from our moms, is that the oocyte is full of mitochondria, relatively speaking, with the tiny little sperm maybe contributing a few of dad's mitochondria into the offspring, but mom's mitochondria outcompete them, and so we don't really functionally inherit any mitochondria from our dads. The only thing we get out of the sperm is the nucleus and the chromosomes, the nuclear chromosomes. So that's a big difference between male and female meiosis. Size. In terms of the cell divisions, you start with a 2N cell. This should seem familiar by now. Synthesis, 4N. Let's do male, is written in the top on the left, so let's do male divisions on the left side. The first division makes 2N cells, and then the second division produces, from each of those 2 cells, 2 new daughter cells are formed. They're each haploid. I already drew this about 3 minutes ago. Review. Each of those will become a functional spermatocyte. That is, they'll develop when it's time. They will each develop a flagellum and be motile gametes. This is not what happens in female meiosis. There are a couple differences. One of them is not so much genetic, one of them is more physiological, so I'm not going to talk about it. That is when during the life of the organism these two processes occur. Males of reproductive age are always making spermatocytes. They're always doing meiosis. Women, when do you do oogenesis? When does meiosis happen in women?
As a baby.
Yeah, it's really young, infant or before, well, infant-ish. Meiosis proceeds and then pauses. So women make a finite number of oocytes, right? You don't make any more as an adult. There's a point at which you run out of producing oocytes. That doesn't happen as much in men. There's an evolutionary reason for that, which is because, in theory, because men make the smaller motile gametes, we can afford energetically to make more of them and waste a lot of them in the hopes that one of them succeeds in making an offspring. But women, because they make the larger, more resource-filled oocytes, single cells, they're so much bigger than sperm, there's a lot more energetic investment in resources in producing those cells, so you don't make as many of them. And that's one of the differences between males and females. Meiosis-wise, though, first cell division is not symmetrical. Up until this point, every time I've drawn meiosis, I've drawn the first division of meiosis, producing 2 daughter cells that are similar sizes. True in men, Not true in women. The first cell division, now grant me a little artistic license, might look like this. Each of those cells is still 2n, so it's still a division from 4n to 2n. But does anybody know what we call that little tiny daughter cell? It's called a polar body. Oh yeah. Google that. Why is this division asymmetric? Because the process of meiosis is loading the eventual oocyte with the cytoplasm and all the resources of the initial cell that starts the process. This is how the oocyte gets to be so much bigger than, in part, this is how the oocyte gets to be so much bigger than a sperm. Then the second cell division does the same thing. Oh, by the way, this is the first polar body or the primary. We'll call it the first polar body. Because what happens in the second division? The second division, the first polar body divides and makes two second polar bodies. And the bigger cell also does, like normal, its second cell division. It produces, now I'm running out of room, so I'll make the oocyte long and flat. The oocyte, the bottom right, that is the egg, and a second polar body. Second PB. So each of the divisions in meiosis in women are asymmetrical. First division makes the primary or first polar body. The second division produces another polar body. So in the end, yes, all four of those cells are N. Each of those four cells is haploid, but the oocyte is huge compared to the others. And this will be important in particular for an exercise I'm going to ask you to do for next class. So the most recent discussion board topic was, do chromosome errors occur during any point? Presumably mitosis or meiosis, since that's relevant to what we've been studying. What happens if the chromosomes don't segregate, don't divide properly into the daughter cells? So most of you found lots of great studies that show that, yeah. Then, the night I was writing this up, after you had just submitted all of those really carefully crafted paragraphs and their references, this was published. 27th September 2019. Last week, this paper got published. Chromosome errors in human eggs shape natural fertility over reproductive lifespan. What do you know, or what have you heard, about causes of aneuploidy, the wrong number of chromosomes in sperm or in eggs? Have you heard anything about that in media, friends, family, life experiences? Have you heard about when you're more likely to have a kid that has Down syndrome, extra copy of a chromosome?
If you wait until you're towards your 40s.
Okay, wait till...
More likely to happen.
And so the answer up here in the front was wait until you're older, say your 40s. So I've heard this, that you're more likely to have aneuploidy, germ cells, sperm or eggs with the wrong number of chromosomes the older you get. That's sort of an idea that's been out for decades. So in this study, they looked at that. They wanted to know, now we're just looking at eggs, not the sperm. They wanted to know, essentially, how does age correlate with the probability that the oocyte has the wrong number of chromosomes in it? Is it true that it's linear, maybe? The older you get, the more likely you are to have an oocyte that has the wrong number of chromosomes. And the quote that I want to share with you from this paper is simple. Aneuploidy follows a U-curve. So they've got age on the x-axis here, down at the bottom, age 0 to 50 years. And the y-axis is a little bit complicated, euploid rate over fertility rate. So euploid rate means the percentage of cells that are euploid, have the correct number of chromosomes, but they've corrected a little bit because when you're 0, 5, and 10 years old, your fertility rate's not huge, so the probability of having an offspring that with an aneuploid egg is low. So they really care about the combination of both how quality are the oocytes as well as how fertile are you at that time. So human peak fertility is, in this range, 15 to 35, say. And they find that the probability of having, if you just follow the human eggs, euploid, the red line, when's your peak probability of having euploid oocytes? Or if you're a guy, having a kid with a woman who has a euploid oocyte. It's about mid-20s. But it falls off in both directions. So it's not necessarily that older is worse. Younger is worse too. Really young women are just as likely to have aneuploid oocytes, eggs with the wrong number of chromosomes, as older women, when you look inside the window of the range of fertility. Which is really interesting for many reasons. First, it's practical to know if you're interested in having kids. And second, it makes us now wonder, why would that be? Mechanistically, what's going on in meiosis that would cause problems both at the start and at the finish of reproductive lifespan, that is. So something to think about. Oh, I want to share one other story with you quickly before we wrap up. 7 minutes or so, then we'll do this exit activity where I ask you to give me all of your feedback on the exam. Feelings, pro and con. We also had a conversation about CCR5 delta 32, this mutation that presumably keeps some humans in Western Europe from getting HIV. And we also talked about, somebody mentioned, I asked, are all mutations bad? Somebody, I think it was in the back, said, well, what about sickle cell anemia? It's a weird situation where if you've got one mutant and one good copy of the hemoglobin gene, then that protects you against malaria. But if both copies of the gene are bad, then you get sickle cell disease. Like there's a benefit of having one mutation, but not two of the same mutation at the same time. And I don't remember if we talked about this bottom point in class, but it was definitely something I was thinking about, is it bad to have, is there any bad aspect of having that CCR5 delta 32 mutation? So yeah, it maybe protects you from getting infected by HIV, but might there be a drawback to having that mutation too?
I read like an article that apparently some scientists in China manipulated these twin girls to have that mutation, and then they were criticized because there is a possibility that that would also shorten their lifespan.
I didn't hear the latter part, but I'm not saying that that's not correct. Somebody probably did hypothesize that. Yeah, so when we get to the genetically modified organisms part of the course, we might talk in detail about the genetic engineering of, it's true, 2 twin daughters in China who were designed to have that mutation. And then we can talk about, is it okay to do that to humans? Should that have been done? And do we know the effects of making these sorts of mutations? Like maybe it's good for not getting HIV, but maybe it's bad for something else. So I just wanted to share this story with you then, because this also just came out in the last couple of weeks, I think. It was right after we talked about that concept in class. I just didn't share it with you at the time, that this mutation, if you're homozygous, these authors say, this is not true, by the way, that's the punchline for the story in the next 5 minutes, is that it turns out that this analysis was flawed. But this was published in Nature Medicine, a pretty high-profile journal, saying that if you have 2 copies of this mutation, yeah, you maybe are HIV resistant, but it shortens your lifespan. And the way these authors did this was, I don't know if it's pretty straightforward or not, but they took a sample of, there's a huge registry of people and their genotypes in Britain, so they had access to these data. And they looked at the number of people 41 years old that had either 1 copy or 2 copies, 1 copy or no copies of this mutation. So they're the heterozygous, so they didn't have the mutation at all. And they saw what frequency this mutation exists in the population. What percentage of the people in Britain have a version of this mutation? And they looked at a population of then 3 or 3.5 decades older people, 76-year-old British people, and looked at the frequency of people that were homozygous for the mutation. That is, both copies of their chromosome had that HIV resistance mutation. And they found that there were fewer people at 76 years old that had both copies mutated than there were at 41 years old, and so that was what led these authors to say, well, it must shorten your lifespan. We don't see as many people living to this age, 76, that have both copies of that mutation. It's a pretty straightforward study to do, but it turns out that some of the math they did, well, it wasn't the math. It turned out that the database that they were using, that they were collecting these DNA sequences from, the genetic information, had some mistakes in it. The data themselves weren't good. So even though the analysis they did were accurate, there's a problem with the DNA sequences that they were using in the first place. So on Twitter, this is the senior author of that paper. The one thing that all scientists fear the most is to find out that a major result you've published was based on erroneous data. That event will affect you for the rest of your scientific career. Somewhat true, but there's been a lot of praise for Dr. Nielsen and the rest of his group that published that paper. Once they found out that it was flawed, they immediately started working to retract it. So we will work with the Nature Medicine editors to get the publication record corrected. So it's bad to put faulty information out there. It's much better to admit it if and when you've discovered that it's erroneous or misleading. So then on the 2nd of October, just last week, a different group, which includes the authors of that original paper, have published a new paper explaining what happened. The only reason I'm telling you all this is because it's important, because a lot of what is important about understanding genetics as citizens or as humans is to be skeptical about what you read and to realize that sometimes you might read the title of the paper and it says something impactful. You have to keep your ear to the ground, that is, keep paying attention to the story because sometimes it changes. Okay, so I'm going to go open up this Socrative feedback. If you'd like to give it, you may. I appreciate it. And then there's one case study from the meiosis chapter in the lab manual to work on for next time. And the next slide, the last slide in this set of slides, is another practice exercise. If you want to, if you've got time for next class, we'll go through that next class. It's a good practice question. Let me go open up this Socrative activity. And here's what we're going to be looking at next time. Why do most siblings look kind of similar, but not identical? Has something to do with meiosis. So thanks for your responses to that Socrative activity. The first 3 are multiple choice. The last 4th question is free response. So if you want to save, if you have any written response you want to give, that's the 4th and last question. Oh, sorry, 6 questions. The last 3 are multiple or free response. And I'll leave this open for a while. You don't have to you can definitely start now, but it'll run past 5 minutes from now. So if you want to take longer to respond, that's fine.